Hello. Today we are going to be talking with Dr. Steffen Fuchs, who is a pediatrician and clinical scientist who investigates the role of alternative splicing and non-coding RNAs in biology and therapy resistance of pediatric tumors, with the aim to translate the knowledge to future RNA-based therapies for young patients. Steffen graduated from medical school at Würzburg University in Germany and received an additional master's degree in experimental medicine. He completed his residency in pediatrics with a focus on pediatric oncology at Charité University Hospital Berlin in Germany as well. In the course of his residency, he enrolled in the clinician scientist program of the Berlin Institute of Health and worked as a postdoctoral researcher in the research group of Professor Johannes Schulte, investigating the role of non-coding RNA class circular RNA in neuroblastoma, which is a form of pediatric cancer. Currently, Steffen is working as a postdoctoral fellow in the group of Dr. Fabienne Mejeto in the Cancer Research Center of Toulouse and continues his work on circular RNAs, aiming to understand their involvement in the development of therapy resistance in pediatric lymphoma. So today we will be discussing his recent PLOS ONE Lab Protocol article entitled Generation of Full-Length Circular RNA Libraries for Oxford Nanopore Long-Read Sequencing and the associated protocol that is posted in protocols.io. So Steffen, thank you so much for being with us and discussing your research.
Thank you, Gabriel, for this very kind introduction. Yeah, I'm happy to be here to discuss our protocol. Thank you for this opportunity.
So just to kick off, can you please briefly explain to us what the protocol is about, how to use it, and why it is relevant?
Right. So basically, we're working on the non-coding RNA class circular RNAs, or in short, circRNAs. So this is an RNA class that basically is characterized by a circular RNA structure, which makes it kind of unique. And it was shown that these RNAs are important for gene expression regulation. And often you find them dysregulated in cancer, which is why it's important to study them actually. So, and when they regulate gene expression, it's mostly by interacting with other RNAs or proteins, which is why it's important to know the whole sequence of those molecules. When you now do classically Illumina sequencing, which is a short read-based sequencing approach, you can detect the circRNAs, for example, by having sequencing reads that fall on the circular junction, so you detect them, but actually the remaining sequence is unknown. You can kind of estimate it, but it's not known. But it will be important to know to understand the mechanism of those molecules. So to get this information, you have to use a long-read sequencing technique, for example, using the Oxford Nanopore platform, to have a read, for example, that spans the whole circle, and then you know what is the sequence of it and where the interactions happen, and this will give you information about the mechanism of action. And this was basically the motivation for this protocol. So, for example, when you want to understand, so you study circRNAs, you want to understand the mechanism of action of a circRNA, it will be pretty helpful for you to perform this sequencing technique, because then you can really study the interactions in depth.
So when you started developing this protocol, can you describe what was the state of the art? In other words, were there similar protocols out there? And if there were, why didn't they work for you? What was the motivation for you to optimize and develop this very specific protocol?
Right. So, I mean, circRNAs itself, they're already known since decades, but only in the last 10 years, people really started to working on them a lot, and a lot of methods were developed. And to really sequence circRNAs in full length, there were just a few protocols out there. And what bothered us a tiny bit about them was that they had, for example, a lot of size selection steps. So, for example, circRNAs, they can be really short and they can be really long. The average length is around 200 to 800 nucleotides. and a lot of published protocols, they use, for example, size selection of more than 1 kb. So you will sequence circRNAs, but maybe the picture you get is a bit biased. And that's why we thought it actually would make sense to design a new workflow that sequences all circRNAs to get really the global picture.
I see. And so the next question that I have now, it's like a two-part question. And so after all the time you invested in developing this protocol, that will work for you. So what was your motivation to first go in the extra mile and have it posted in a public platform? And second, after you did that, you went and had the protocol peer-reviewed and published as an index paper. So what were the motivations for you to take these two steps that are time-consuming?
Yeah, that's true. So the first question, why to publish the protocol? I mean, I know that there were some platforms out there where you could basically post a protocol, but then it was like a fixed document. It could never be edited, which is maybe less interesting. But then the approach of Protocols.io to really have a protocol out there that is actually kind of live, you can edit it, you can post new versions of it. That sounded pretty interesting to me. But I didn't know about that platform at all. And actually, I learned about it by an online course that was actually taught in Berlin by the Quest Center for Reproducible Research. That's how it's called, of the Berlin Institute of Health. Since they have a collaboration with Protocols.io, and there they teach us actually, okay, you can produce a protocol that is then published online, which will be actually helpful for the research community. And then you can actually get it published in a scientific journal. And this seemed to be like a really great approach, and also like to get it out there to really help the fellow researchers. And then the next question about the review process itself. Well, for us, it was pretty helpful. We had 2 reviewers, and they really seem to be specialists in the field, also on circRNA research, which is pretty a niche. The comments were mostly going into the direction of be as detailed as possible. So you work on a workflow like for months or even a years. So for you, it might be pretty clear, but maybe not for the other researcher out there that tries it for the first time. So you really have to go into more detail. And then also describe the workflow from A to Z. So for example, with our protocol, basically it's a method to generate a sequencing library. So to generate the data, we didn't work on a bioinformatics tools to analyze it, but the reviewer said, okay, maybe it makes sense to give a recommendation also to analyze the data. And I think the reviewer was completely right. So now we also describe this in complete. So you have a holistic approach, you get how to create the data and how to analyze it. It's pretty complete. And then the last comment, which I find is maybe the most important one, the reviewer also suggested to actually validate the circRNAs that you sequence by a different approach. So for instance, we sequence them, but then we now use, for example, PCR and Sanger sequencing to validate them by a different method to see that they were really correctly sequenced. And like this, it's I think really thorough.
So if we can summarize, the feedback that you received from the reviewers were in the sense of making the protocol, the reporting of the protocol, comprehensive, detailed, and validated.
Correct, absolutely correct. Yes.
And once you had those reviewer comments, what motivated you to make the peer review history public together with the paper in PLOS ONE?
Right. So I think actually for a scientist that reads papers, to see the peer review history is actually pretty helpful, because you see what were the comments in the first place and how did the researchers approach them. I know that a lot of times this is not public, but actually I find it pretty interesting, and that's why we decided to share them, because yeah, the people see the final product, but actually what were the important questions in between. I think this is really enlightening and can help to increase the quality of research.
And I would like us to change gears a little bit and go deep into the protocol itself. Yeah. And I would like to know during the process of optimization and development of the protocol, is there anything that you tried but didn't work and didn't make it to the final version of the protocol?
So circRNAs itself, they're pretty lowly abundant. So this is why you actually have to have a lot of starting material. So this protocol was developed with cell lines, so here you don't really have a limitation. So for instance, we started with 7 micrograms, and this might be actually a limiting factor for clinical tissues, for example, or so. So actually, one thing we're working on is actually to lower the amount of input material. This is currently ongoing. And then we also want to use this protocol also to sequence, for example, tumor tissue, which we didn't try before, to really make it even more generally applicable to other tissues, not only to cell lines, but also to clinical tissue and so on. And this was so far not in this version that is published. Since this is ongoing, we didn't test it enough, but a future version will come.
And those are the lines you're working on, minimizing or reducing the amount that you need of starting material and working on more biologically relevant samples, not set lines. But has there anything within the protocol, have you encountered any issues that you have had to troubleshoot?
So maybe I can go a bit more into the detail, because as I said, there were already published protocols out there, and they're also great. We also tried them, and they worked in our hands. But as I said, one factor that was a bit complicated for us was the size selection that we changed. So actually, in our protocol, we don't select for only long RNAs. We take everything into account, basically. And then also we tried the published protocols, and the sequencing output was a bit limited in our hands. So we used several actually measures to increase this. For example, we used more input, as I said. We tried to increase for circRNAs stronger. So for example, during the troubleshooting, we used different methods to digest linear RNAs. You can do this enzymatically. You can, for example, use a selection for polyadenylated RNAs, and then you take all that are poly-A out. So you have the circle that has no poly-A tail, and then you keep it, which is then another enrichment step, for example. Really important is also a really well-working ribodepletion, because more than 80% of your RNAs in a cell are actually ribosomal RNAs that you would also sequence if you don't digest them. But in this moment, you're not interested in them, so it makes sense to have a really good ribodepletion. So we tested different approaches for this. We tried to use different number of PCR cycles, because of course you want to have enough library, but you also don't want to overdo it, to have too many PCR duplicates and so on. These were actually things that we did during troubleshooting. Other things that we found really important were also actually quality control steps. So when you do your library and you actually enriched for circRNAs, you actually kind of have to see if that worked. So library validation can be classically done also for Illumina by using something like bioanalyzer, tape station, or fragment analyzer to see that the library has the correct size. You can quantify the library by qubit, for example. But actually what we also used was qPCR for example, selected circRNAs to see that they were enriched. And then also using primers, for example, for 18S or 28S ribosomal RNA, and also other RNAs to see that they get depleted, which is important because otherwise you will sequence a lot of RNAs, but maybe not the ones that you are interested in. Yeah.
And can you now summarize, what do you think of the current protocol are the most critical steps? I mean, you have mentioned some, but can you, put them together in a collection of critical steps for the user?
Right. So I think since they're so abundant, for me, the first critical step is that the ribodepletion is working well. Otherwise, you have a lot of sequencing data, but maybe a lot of ribosomal RNA and not the circRNAs. Then the circRNA enrichment has to work well. And this is why the QC in the end, by doing qPCR for circRNAs, then also for ribosomal RNAs and so on, is really important for me. If this doesn't work well, if you don't see an enrichment here, then you shouldn't sequence the library. Then just save your flow cell and do it again. Then after you have your cDNA library, you actually have to create the actual nanopore library. So you have to add the sequencing adapters and so on. And here it turned out it's also pretty critical to keep the correct temperatures in the PCR cycler. So for instance, you have to ligate PCR adapters and you think, okay, you do it, no problem. But we had one problem with a heated lid of a thermocycler that was not correct. And then the library preparation failed. And we thought, okay, what happened? And then we used a different machine and it worked perfectly. So even these little details can be important. And then when you perform the final nanopore sequencing, you should be careful, for example, to overload the flow cell and keep a close eye on the sequencing process itself, because there can be also some things that can go wrong. For example, you can overload the flow cell, you can have air bubbles in it. But if you see this, you can do something about it. You can, for example, wash the flow cell, reload the library. So this is also a critical step.
And now, moving back a little bit and heading towards the end, can you tell us what has been the significance of getting this lab protocol peer-reviewed and published for your career as an early career researcher?
Right. So we adapted this protocol and published it. And at the moment, it's just a method itself. But now we're applying this method actually to research questions. So for example, we are, as you said in the beginning, we're studying therapy resistance in a pediatric lymphoma. And now we want to use this method to analyze a cohort of, first of all, resistant cell lines to see if we find circRNAs that are linked to resistance. And then in the next step, this is why it's so important to improve also the protocol, for example, lower input and so on, to analyze clinical samples and to see if we also find circRNAs that are linked to therapy resistant in actually clinical tissues. And this will then hopefully also create new publications. And as you said, it's really important for an early career researcher to kind of build your track record to get the whole career going right.
And before we wrap up, is there anything that you would like to mention that we haven't touched in this conversation?
I mean, I think we covered pretty a lot of the process. Yeah, I think the whole thing itself went pretty well. Now I would actually really like to see that other researchers using this method and maybe also comment it, because I saw on protocols.io, you can actually also comment the protocol, you can ask questions, so it's still interactive. I actually commented also some other protocols. I find it really great, and I'm really happy to interact with other people that are using this technique.
Well, Steffen, thank you so much again for carving some time to talk to us. And yeah, thank you so much.
Thank you. It's my pleasure.